It's a great pleasure to come here. I first came here in 2010 for the first Crick Symposium. At that time, there was a church. You guys can see across the bridge there is a church and nothing else. There is this hotel, the church, and the rest is marshland next to the lake. Now, every time I come here, new buildings coming up. It's just amazing how the last 15 years, probably because of Cold Spring Harbor Asia, you sort of laid a foundation that people want to attract people to come. So it's great to see all these development. And thanks to the organizer to allow me to speak a little extra long, for 15 minutes longer. So I'll not waste the time and start doing it now. Let's see. So our lab studies how neural circuits are assembled during development and how their functions, how their architecture enabled them to perform specific functions. And we use flies and mice as our model organisms and also develop tools so that we can answer these questions with increasing precision. And as Bo Lee mentioned, I wrote a textbook which feedback, focus on how discoveries were made and, of course, have a lot of influence to our own research program. So today I'm going to talk about our work in neural development. In the first part, wiring specificity of the fly olfactory circuit, and in the second part, the wiring specificity of the mouse brain. So the modern study of the modern wiring can be traced back to Roger Sperry's chemoaffinity hypothesis over 60 years ago. Based on his observation that regenerating retinal ganglion cell axons target specific regions based on their position in the retina, Sperry speculated that cells and the fibers of the brain and the cord must carry some kind of individual identification tags by which they are distinguished one from another almost in many regions to the level of the single neuron. The chemoaffinity hypothesis has inspired generations of developmental neuroscientists to identify molecules that guide axons to targets with spectacular success, for example, summarized by this nice review. But still, we still do not know how these different cell surface proteins work together to assemble a functional circuits. Furthermore, if Sperry were correct in his last phrase, to the level of the single neuron, which I highlighted here, we would have a wiring specificity problem. We know that a human brain contains about 100 billion neurons, making more than 100 trillion synapses. But a human genome only contains 20,000 genes, and about 3,000 of them encode cell surface proteins or identification tags. So there are far fewer number of cell surface proteins than the number of neurons and the connections to specify to the level of individual neuron. Even the fly brain contains orders of magnitude more neurons and the connections than there are cell surface proteins. So in the process, in the last 25 years or so, we are studying how these different cell surface proteins help wire the brain. But in the same time, we also come up with some interesting solutions to this wiring specificity numerical problem that I will summarize to the end. So I will first start with fly olfactory circuits. So in a fly, odors are smelled by these olfactory receptor neurons. Like in mammals, each olfactory receptor neuron expresses one odorant receptor. Neurons express the same receptor, project their axons to the same one of the 50 or so glomeruli on each side of the brain. Their information is relayed by second-order projecting neurons, each of which send dendrites to one of these glomeruli and send axons to high brain centers to mediate innate and learned olfactory behavior. So in the wiring of the fly brain, each ORN targets axon to one of the 50 discrete glomeruli. Each PN targets dendrites to one of the 50 discrete glomeruli. And the 50 types of ORNs and PNs form precise one-to-one synaptic partner matching. So we've used this system in the last 25 years trying to decipher, as a model, to decipher the general principles of how neural circuits get assembled. This complex enough so that we hope the lessons we learn can be generalized to even more complex system. So summarizing 25 years of work on one slide, in this one-to-one connection, being made in adult, we find that actually it's a stepwise process. In the beginning, there is these projection neurons that send dendrites to innervate the antenna lobe first. Based on their lineage and birth order, they set up programs. And then olfactory receptor neuron axon first invade to antenna lobe, they come to the surface of the antenna lobe, and then they try to find their partners. And we identified many molecules that regulate these different events. But one of the common things is that when we delete any of these molecules, even with a null mutation, we usually have a partial phenotype that most of the neurons still target to the right glomerulus, and there are some mistargeting. Because the wild type is so precise, we can always see, okay, this is very important. So that implies there is a lot of redundancy, which then leads to robustness. It's fine for the organism, for like scientists who want to understand thoroughly how this program is run. We encounter the problem of how one can delete one gene at a time to invest this problem. So an alternative approach is to re-engineer cell surface code such that XORNs, instead of connecting with XPN, now connect with YPN completely. If we can address this question, we can claim that we have reached a satisfactory level of understanding. Like the famed physicist Richard Feynman used to say, what I cannot create, I do not understand. So in the first part of my talk, I'm going to focus on one unpublished study to address this rewiring problem. Before doing that, I'll give you 3 snippets of recently published work to provide enough background. So this, the first is dimensionality reduction. So if you look at adult, each one of the ORN needs to select one of the 50 glomeruli in a 3-dimensional structure. That's a pretty complex task. It turns out that during development, if you look at projection neurons, whether they are invading and surface or the interior, they all have dendrites extended somewhat to the surface, so that because ORN axons come to the surface, so this 3D search problem becomes a 2D. Furthermore, if you look at an individual ORN type, their axons actually move along a 1D trajectory, about 30 degrees out of 370 degrees of the surface. And this trajectory matches precisely with their partner dendrites. So in a way, the 3D problem can be reduced to a 1D problem, that synaptic partner matching can be achieved by axons moving a genetically programmed trajectory that only encounters a handful of glomeruli instead of 1 out of 50 in a 3D. So within this 1D trajectory, you still need to find which one of the 5 to 8 glomeruli you are going to encounter. Sorry. And this problem was cellular mechanism was deciphered by both my graduate student Chuan Yunxi and a postdoc Tong Cao Li, who is now in Zhejiang University, and he's going to give a talk tomorrow. So it turns out that these axons actually send branches all along the entire trajectory, touches all type of projection neurons, but only those that are matched with the postsynaptic partner become selectively stabilized, and the rest withered away. So that suggests that there is both potential attraction and repulsion there. So the third piece of data I want to share with you, this was done actually over a decade ago by a graduate student, Wei Zihong, to identify the first molecular regulators of this wiring. He identifies through genetic screen 2 molecules called tannurins, 10M and 10A. They express in exquisite expression patterns. For example, low for 10A, high for 10A, this glomerulus, and high for 10M. And in fact, when you look at the ORN PN, they go to the same glomerulus, they are partners, then they express matching level tannurins. High for 10A, high for 10M, high for both, or high for neither. And the genetic analysis indicate that they mediate homophilic attraction to instruct wiring. By the way, Wei Zihong has now become a full professor in UCLA, tenured, of course. So working on tannurins, get tenured. So right now, okay, so I'm going to switch to now the Cheng Rui's work on rewiring. He's coming soon to set up his own lab in West Lake University. So his job was to rewire DA1 ORN to VA1 VPN. In fact, he did a bunch of them, but I'm going to focus on this particular one, DA1 to VA1V. How does he do that? So if you look at, so he starts with a driver, GAL4 driver that labels only DA1 ORNs. So this is in adult, this is in pupa. During the time they are making synapses, you see that they form more branches here, but they also form branches elsewhere. And they form branches more towards at the postsynaptic target and much less in VA1V. And that's the reason why in adult, if you look at DA1 ORN's axon compared to DA1 PNs, nicely match. VA1 VPNs do not match at all. So we have these cell type specific marker that allow us to do quantitative analysis. So in principle, if we switch DA1's ORN cell surface protein to that of VA1V ORNs, then we can do the rewiring, because then you can presumably utilize this VA1V ORN cell surface protein. So that's actually the incentive behind us spending a lot of time studying, doing single cell RNA sequencing studies, published in these studies. And we find that even for these very closely related ORNs, they differ by many dozens of differentially expressed genes in the cell surface protein. So that's a lot of work to even for flies to find out. And nevertheless, Cheng Rui recruited a graduate student, Zhuoran Li. They also collaborated on the dimensionality reduction study. The genetic screens to look for these molecules, and they identified 10 such molecules. Some of them mediate homophilic attraction, for example, tenurin A and tenurin M, we found out. And the other two actually were known from these studies to mediate homophilic attraction. We find that they expressing matching partner PN and ORN. They also found a bunch of heterophilic repulsive molecules that have an anti-matching expression. So armed with these 10 molecules, now Cheng started to do genetic manipulations to change, just express this on DA1 ORNs, molecules, either knockdown or overexpression, to trying to switch its profile to the VA1 VPN. So this is the beginning. Wild type or DA1 ORN matches with DA1 PN. None of them match with VA1 VPN, shown here in this diagram that image that I already showed you. So it turns out that if you change the profile of individual molecules, sorry, this is not, maybe I use this. So there is very little change. One molecule does not do much. If you start to do combinations, you started to see partial switching. So blue, we want to switch to pink. And finally, when we did 5 different molecules, we did a complete switch. The green ORNs now match with magenta PN. Mission accomplished. So what are the molecules? What can we learn from this switch? This is actually a lot of work. It turns out that there are 3 things you need to do. First, you need to match attraction. DA1 ORN, we want to match attraction to target PN, to the VA1 VPN. And therefore, we need to downregulate these 2 adhesion molecules because they are expressed in DA1 PN but not in VA1 VPNs. Second, we need to change repulsion. Decrease repulsion to the target PN. So this PTP10D expressed in DA1 ORN make it be repulsive to VA1 VPN. So we need to knock it down, and we did that. And the third is we need to increase repulsion to the original PNs. So it turns out DA1 PNs express these 2 molecules called Philly and CAC1. So we overexpress their partners to make them more repulsive. And after 3 of these, we can now achieve the repulsion. And then we also utilize the same principle to rewire this VA1D OIN, the different type, to 3 different partners. One of them with full success, and the other 2, at least more than 50%, becomes rewired. So these 3 principles can allow us to precisely rewire at least 2 of them to completion and 2 of them majority. So for now, I'm going to focus on this last one. This the first one I talked about, VA1D OI and VA1B PNs. After rewiring, you see actually it only occupies half of the magenta dendrites, because the other half is presumably innervated by its endogenous OIN partner, because all our genetic manipulations were done on these green OINs. So physiologically, indeed, we see that if we give them an odor that normally activates VA1B OINs, it will activate both wild type and rewire the fly. However, if we switch them to an odor which normally only activates DA1 OINs, in wild type, actually, there's an inhibition because of lateral inhibition in the olfactory circuit. Now it is switched to excitation. So physiologically, indeed, the rewired connections are physiologically relevant. Now I wanted to introduce what are these odors. This PA odor is actually a cuticular pheromone that promotes male courtship, whereas CVA is a male-specific pheromone that are mating males to inhibit male from mating with other males. In fact, when flies mate, in addition to give female sperm, males will inject some CVA so that mated female smells like male, so that other males will not compete with their sperm. So this prediction is that if we rewire inhibitory pheromone sensory neuron to a PN that normally promotes male courtship, you would predict that males will find other males attractive. And indeed, that's the case. So this is a behavior of a rewired male put into a normal male. Together, they will chase after it and singing courtship song. So the flap of one wing, unilateral wing extension, is a typical courtship song behavior. And this normal male feels strange, start to run away, but it's being chased vigorously after, singing courtship songs. Here's a snapshot of 3 different courtship song ritual. And one day, Chen found some extra males. He put 5 of them together, rewired males. They will chase after each other, forming courtship chain, one after another. One is being courted from by the fly in the back and trying to court another one in the front. So this is a courtship chain. One of them was flapping its wing. So just to summarize the first part of the fly talk, dimensionality reduction simplifies synaptic partner matching from choosing 1 out of 50 targets in a 3D space to 1 out of a handful of targets along a 1D trajectory. And synaptic partner matching is achieved by a combination of homophilic attraction between synaptic partners and heterophilic repulsion between non-partners. And by breaking the combinatorial cell surface code, we can now switch synaptic partner with precision and alter physiological behavior in a predictable way based on their normal functions. So I'll move on now to the second part, the mouse work. And this was actually inspired by our studies about tangerines in the fly that matches pre- and postsynaptic partners. That was one of the first synaptic partner matching molecules identified. So we wanted to know, because these tangerines are highly homologous in mammals. In fact, humans have 4 tangerines, mutations in any of which will cause different kinds of neurological diseases. So we want to test whether the mammalian tangerines play similar functions. So this first story started by Nick Burns, my former graduate student. He focused on one of the tangerines called 10-3, which is highly expressing a very interesting pattern of a part of proximal CA1 and distal subiculum. And from previous work, we know that there is a topographic projection. Proximal CA1 neurons project to distal subiculum, middle to middle, and distal CA1 projecting to proximal subiculum. So 10-3 high neurons connect with 10-3 high targets, just like the whole fly olfactory system, ORNP, and have matching expression of tannurines. So Nick made a conditional knockout and tested and showed that tannurin is required both in CA1 axons as well as in subicular target to mediate this precise precision targeting. So because this work is published, I'm just going to summarize that tannurin instruct this wiring also through homophilic attraction, because he was able to show that they can mediate homophilic attraction by causing aggregations of cells, non-adhesive cells. So this raises the question, what controls the distal CA1 to proximal subiculum? We hypothesized that there may be other molecules that are expressed in inverse patterns that mediate these kind of connections. And postdoc Dan Pedrick and Yang Ray performed single-cell RNA sequencing to look for molecules that have anti-correlation. Again, to cut a long story short, because this work was published a few years ago, we found a molecule called latrophilin 2, or latrophilin 2, LPH2, that expresses exactly in counter inverse. For example, they are expressed only in distal CA1 and in proximal subiculum, inverse to tannurin. Unlike tannurin, they do not form homophilic attraction, but tannurin and latrophilin bind to each other extracellularly. So heterophilic binding but inverse expression immediately suggests to us that they might play a repulsion role. And indeed, we, through a lot of genetic studies, loss and gain of function describing this study, we found latrophilin 2 can repel tannurin expression, so tannurin expressing cells. So when proximal CA1 comes, they first encounter the subicular field and being repelled, and then when they enter the distal subiculum, they are attracted. So this both the push by latrophilin 2 and a pull by tannurin 3 explains why these magenta axons target to magenta target. And in addition, latrophilin can also work on the axons, and using 10-3 as a repulsive ligand to guide these green axons, so that green axons only target to green area but not invade magenta area. So this mutual repulsive, reciprocal repulsion enable these two different class of neurons to occupy, have a targeting specificity within these two regions. So now come to some of the more recent studies. I just told you that tannurin and latrophilin mediate proximal CA1 to distal subiculum and distal CA1 to proximal targeting. But turns out that they are expressed also in entorhinal cortex. And medial entorhinal cortex that expresses very high tannurins have reciprocal connection with proximal CA1 distal subiculum. Lateral entorhinal cortex, which has very high expression of latrophilin, on the other hand, connect with distal CA1 and proximal subiculum. And these two subcircuits have been hypothesized by physiologists to preferentially represent object and space. And more strikingly, subiculum also project axons to thalamus, hypothalamus. In these targets, they also have these tannurin latrophilin counter gradients in both messenger RNA and proteins. And from our tracing studies, it turns out that also they follow this tannurin to tannurin, latrophilin to latrophilin Luo, likely organized by the mechanism that I just described to you, homophilic attraction and reciprocal repulsion. And in fact, graduate student Alan Gingrich spent last few years test these ideas, whether these 10-3 latrophilin to play a role in establishing these other connections, and do they use the same mechanisms. And I'm going to, because of time, I'm going to show you just one example of entorhinal cortex connecting with subiculum, distal subiculum, and proximal CA1. So what Alan did was to inject Cree, lentivirus Cree at P0 as soon as the pups are born into medial entorhinal cortex. He injected enough so that about half of the cell have the Cre, half does not. And then in adult, he should inject 2 different kinds of AAVs. One of them expressed only in Cre off cells, which is a wild type. And the other one is on Cre on cells, which is a mutant, if they are injected into the flox animal. Of course, when they are injected into wild type, everything is wild type. So these are the injection schemes, and then we can trace green and magenta axons in adult and compare them in the same animal. If you do them in wild type, you see that green and magenta are completely intermingled. If you inject in medial entorhinal cortex, they project proximal CA1 and distal subiculum. Quantification here for the subiculum. They are highly enriched in distal subiculum. However, if we inject them into 10-3 flox animals, now green becomes mutant, and they will spread more proximally in subiculum and more distal in CA1 into the lateral filling 2 regions. So indicating that lateral filling, this 10-3 is highly is important for this specification in axons. Acts autonomously as a receptor in medial entorhinal cortex axons. Then she also tested whether target require these molecules by deleting lateral filling attenuring, utilizing a Cre driver that's only expressed in subiculum, but not in CA1, nor in medial entorhinal cortex. So if you do this as a control without a Cre into the double flox animals, if you inject into medial entorhinal cortex, they target to distal subiculum and the proximal CA1. But when she did this in conditional knockout, only in subiculum, you see subiculum has a diffusion, and CA1 is fine because CA1, nothing is deleted. And shown in the quantification here, CA1 completely normal. In the same animal, subiculum, you have a spread, phenocopies, the axon deletions. So to cut a long story short, Alan examined many of these connections, and they found this 10-3 lateral filling 2 is indeed repeatedly used, follow the same logic that we described originally in proximal CA1 to distal subiculum. Homophilic attraction and reciprocal repulsion. Additionally, we found that, for example, in all of these experiments add together, one molecule can really do many things. So we call it multifunctionality. For example, 10-3 in distal subiculum, at the same time, it can serve as attractive ligand for 10-3 positive proximal CA1 axons. It can serve as a repulsive ligand for lateral filling 2 CA1 axons. It can serve attractive ligand, repulsive ligand for medial and lateral entorhinal cortex axons. Besides their role as a ligand, it can also serve as a receptor for its own axons to target to memory nucleus in the hypothalamus. Attractive receptor for 10-3 and repulsive receptor for lateral filling 2 target areas. So really, this repeated use and multifunctionality enable these 2 molecules to instruct targeting of many different brain regions. We wanted to see whether this kind of molecular cassette that we just described is even more widely used in the brain. So in the last few minutes, I'm going to describe to you a few experiments to look at 10-3 lateral filling 2 across the whole brain. Do they exhibit inverse expression patterns beyond the hippocampus? And do the circuits follow this 10-3 to 10-3 lateral filling 2 to lateral filling 2 rule? And whether 10-3 lateral filling 2 play a role in enforcing this connectivity rule elsewhere in the hippocampus. So to do these experiments, we actually made conditionally tag the mouse. I'm going to show you, first of all, just tag the 10-3 with HA, lateral filling 2 with GFP, and this is a whole brain light sheet microscopy imaging. We looked at many different stages, but I'm going to show you a P2, a postnatal day 2 experiment done by Yuri Chong and Dan Pedrick. So you are going to see a brain which is imaging from top to the bottom. From top is cortex and the superior colliculus. 10-3 is in magenta, lateral filling 2 in green. You see that there are inverse gradients. Superior colliculus in the cortex. In the cerebellum, I'm coming back to. In the striatum. more striatum. This is the mammary nucleus and auditory brainstem. So I'm going to give you some examples in 3 different systems. We actually have examined more because of the time. I'm going to just, so I started the talk by telling you about retinotopic projections, the classic Sperry experiment that led him to propose the chemoaffinity hypothesis. And indeed, about 30 years after his proposal, we know a lot. Work from John Flanagan, Friedrich Bonhoeffer, and others have identified, ephrine and EPH play a very important role in mediating retinotopic projections along the anterior-posterior axis. Relatively speaking, much less is known in the orthogonal axis what controls dorsal-ventral. So it turns out that tangerine is highly expressed in ventral retina, lateral in dorsal retina, and tangerine is highly expressed in medial and superior colliculus, which is similar to the tectum in a non-mammalian vertebrate, and lateral too is in the lateral subsuperior colliculum. And we know from this classic work that ventral retina project to medial superior colliculus, dorsal retina to my advancing is not as smooth. So this follows very nicely the 10-3 to 10-3 lateral to lateral rule. So the next example is auditory system. So auditory, one of the key organization principles is the tonotopy. Sounds of different frequencies are detected by hair cells in different positions in the cochlea, and then they have formed these topographic projections to the cochlear nucleus, to brainstem nuclei, to inferior colliculus. Everywhere you see, there is tonotopy along a particular axis. But very little is known what kind of molecules regulate this tonotopy and whether the same molecule or different molecules are used. It turns out that if you look at 10-3 lateral to everywhere, every nuclei we look, 10-3 lateral to form inverse gradients. And every place we look, 10-3 is always in low frequency zone, and lateral 2 is a high frequency zone. So you can imagine low frequency should project low frequency high to high, so therefore 10-3 to 10-3, lateral 2. So Dan Pedrick is now just starting his own lab in Johns Hopkins. He's going to focus on studying how using 10-3 lateral 2 as an entry point to study how tonotopy is developed, and among others. In the cerebellum, we have this famous Purkinje cell that form a single layer. You can see that 10-3 lateral 2, 10-3 lateral 2, 10-3, they form these alternating bands. And these projected Purkinje cells are going to project to cerebellar nuclei to the rest of the brain. In the cerebellar nuclei, you also have 10-3 lateral 2, 10-3 different zones depending on the section planes. Here, unlike the retinotopic or tonotopic map, we know less about which part of Purkinje cells project which part of cerebellar nuclei. So we actually developed a tool that enables us to examine this with more precision. We conditionally tag 10-3 with HA, flanked by followed by stop and then FRT, so that in cells that express FRT, 10-3 protein is labeled with halo. So now we can look at 10-3 made from different cell types in the same animal with different colors. For this example, we are injecting lentivirus flip into the cerebellar cortex to turn 10-3 HA into 10-3 halo in Purkinje cells. And then we can examine halo-positive Purkinje cells in cerebellar nuclei, which 10-3 is now only labeled with HA. So here is a cerebellar nuclei. This is the neural cell. These proteins are made by cerebellar nuclei. And you see that there is actually lateral 2 is complementary, whereas there is no 10-3, lateral 2 is very high. Now, if we look at axons from Purkinje cells, they only go to the 10-3 HA region and avoid lateral 2. So 10-3 project to 10-3. This is in wild type. Now, if we delete this in lateral 2 mutant, then the 10-3 positive Purkinje cell axons will now invade the lateral 2 positive regions as quantified here. So indicating that not only these molecules outline these circuits, connecting circuits, they are also important for instructing the precision of these circuits. So just to last slide to summarize what I just told you about, we utilize the fly olfactory circuit in mammalian, hippocampus, and others to illustrate, going back to the problem, how does a limited number of proteins help wire many neurons and connections? So some of the principles, combinatorial cell surface code, play very important role. These 10 molecules that I just described in the first part can be repeatedly used to instruct multiple connections just by different combinations. And they can also be executed in a stepwise manner. For example, you first choose a trajectory with a set of trajectory molecules, and then the trajectory molecules can be used in combination with the synaptic matching molecule in different parts, reuse the synaptic matching molecule in different parts of the trajectory. This part still requires more experimental test. And last few minutes, I told you about this reuse of ligand-receptor pair, and also the multifunctionality of these molecules across multiple nodes of a network and potentially many networks really shows you how just with one pair of proteins you can help instruct many many connections. So good timing. This is an acknowledgement slide. The work I told you about in flies mostly were done by Cheng Ru, and also Zuran Li made a lot of important contributions, including identify repulsive molecules that enable this combinatorial coding that I don't have time to talk about. And the work in the mouse was done mainly by Dan Pederick, Alan Gingrich, and Yuri Chong. So thank you for your attention. I'll be happy to take questions.
Thank Liqun for the great talk.
You are not student yet.
Students first, student postdocs. Maybe Yang can ask first while students
Well, Yang is young, look young enough. I forgot to say that, Louis has piano. I wish every talk I have will be preceded by the beautiful Chopin. Really put everybody in the mode. So thank you.
All right, so I'll pretend to be a student. So what about the visual system? So you showed the retina to superior colliculus, but what about the thalamus and thalamus to cortex, LGN?
They are expressed in an interesting way too. Just retina topic is the easiest to show. So many different. So by the way, afferent EPH, you probably know the afferent A EPH has been hypothesized because their expression pattern in retina, in the thalamus, in the superior colliculus and the cortex, they have been hypothesized to be reused, but never shown by any experiments. And in fact, of all the papers on afferent EPH, I'm not trying to stamp on if afferent people's toe, but it was 20-some years ago. All the experiments were done with full knockout, not conditional knockout of axons targets, except one experiment. So they really have, in our, Alan Gingrich, in one paper, she did 12 different conditional knockout to really pinpoint the role of, dissect the role of axons in targets. And I didn't have time to go into detail. There are actually some interesting variations about the mechanisms.
Students, postdocs?
Yes. Yes, very good question. We are starting to examine that. So, these molecules are used repeatedly in forebrain, midbrain, hindbrain, actually spinal cord. One of our collaborators also found that they are important for spinal sensory neuron projection to the dorsal horn. Anyways, whether they are regulated by the same type of transcription factors or mechanisms or whether they are different. My expectation is they are going to be different because these different regions have different transcription factor code. And indeed, our limited studies by looking at a single cell RNA sequencing data during early development, when they are these proteins have graded expression to see what transcription factors correlate with 10-3? What transcription factor correlate with lateral filling 2? They are very different. In every brain region, they are mostly different. So my guess is they are just utilizing whatever they can, to create these patterns. By the way, it also says that, the repeated use of these molecules, you can imagine that it's possible that it's easier during evolution to evolve transcription regulation of the same gene, because these molecules work very well at the cassette, homophilic attraction, reciprocal repulsion. Once you figure that out, it's easier to evolve expression patterns than to invent more molecules. And in fact, lateral filling 2 and 10-3 are the most gene sparse region, in a way. That lateral filling in the 10-3, if you look, where's the next genes? They are hundreds of megabases away on each side. So they probably harbor a lot of regulatory elements.
Thank you. Okay. It's really beautiful. And so assuming these molecules tenuine and lateral filling, they are membrane proteins. And
Yes.
I was surprised that there are a lot of long distance kind of connections kind of regulated by this.
Yes.
And
So maybe I should correct the wrong impression just once. So we think these molecules are not, for example, attract CA1 axon to subiculum. What they are doing is, once the axon reaches the subiculum, which subregion do they choose? Everywhere we look, it's the same thing. For example, tonotopy from brain region 1 to brain region 2. There must be other molecules. But once you get there, whether you go to high frequency zone or low frequency zone, so they are really serving as a role as a target selection.
Okay, that makes sense.
Yeah.
Thank you very much. It's very, very impressive work. I wonder, you told very nice story during the development. I wonder, do these proteins are needed maintaining the synapses?
Yeah, that's a great question. We looked at these developmental studies. Most of the brain regions we looked at, it's very transient. The peak of the protein expression, actually in the global study, we did a quantification. The peak of expression coincide with the time when axons reach the target to try to select which region, which subregion to make target. The only exception is the hippocampus, where we can still see in adult the early specific pattern. Proximal CA1 is high in 10-3, distal CA1 is high in lateral filling. But even there, the magnitude of expression is much reduced. So at least their primary role is in many places during development. Whether they play a maintenance role, for example, in hippocampus, it's possible. In fact, another multifunctionality of these molecules, my colleague Tom Sudoff studies these proteins for synaptic organization. Some of them, he does not study the exact pair, but he studies one pair with another lateral filling or one tenuine with another lateral filling. So in those cases, they could be used for maintaining synaptic synapses or organization.
Just a follow-up, when you refer to expression, it's at mRNA level or it's a protein level?
Well, I'm referring to protein level.
Yeah, okay, thank you very much.
Yeah, because all the quantification was done with the whole brain, with these proteins, tagged proteins.
Thank you for the great talk, and my question is a little bit similar to him. That I'm curious about the plasticity.
Yeah.
I think, as you say that as mice or flies become adults, they cannot really acquire such rewiring ability. So I'm wondering what you think is responsible for the loss of the plasticity. Is that because the axons cannot reach to the other place?
Or these proteins turn off.
Just lost.
If these proteins don't express, then you cannot find a target. But I should say that sometimes the injury can induce plasticity, but sometimes it's known that injury of neurons can sort of cause the clock turn back to make these neurons as if they are young neurons again. So they might be able to re-express these proteins.
So even
but then you need to still find a target. Hopefully targets still have these proteins.
Okay. So, and even though the target is far away, they can still.
Yeah, so as I mentioned, even the target is far away, these molecules don't help you to get to the target. They help you to select which subregions. But a lot of injury will cause sort of a reprogramming of neurons so that some other cues that lead them to the target may be re-expressed. So.
Thank you so much.
Sure. Song Hai.
I have a question. In the mammalian system, do you think between CA1 and the speculum there is a single cell resolution connectivity?
Yeah, actually, that's a good question. So to what extent these tellurines specify, whether there are further molecules that regulate target. So I should say for proximal CA1 to distal subiculum, tellurine expressing regions are small, as you see. If you look at a single axon of CA1, proximal CA1, it actually occupies pretty much the entire zone of lateral filling, distal subiculum zone. So my guess is that further specification are experienced. Molecularly, this is it. But the other side, distal CA1 to proximal subiculum most likely have additional molecules because they are big. By the way, everybody asks mouse questions. You forgot about flies. Okay, David.
So in the fly,
yes.
How do you achieve the dimensionality reduction so it only touches a few glomeruli?
Yes. So it's all genetic precision. So we think a set of molecules, we found one, but there is more. A set of molecules, when the axons come in, they have this 270 degree of surface to move. And they only choose 30 degrees. So there must be a set of molecules that tells them which of these parts are going. So one of my new students, Jordan Klein, who I didn't contribute a little bit, her thesis is going to try to find the molecules that cause them to choose trajectory. And so far, all our rewiring experiments were done for OR and PN that target similar trajectory. So we only need to change their synaptic molecules. But if we find the trajectory code, then we can actually change the trajectory and synaptic partner so that we can have a more complete wiring. So for now, it's like pheromone to pheromone. But the other trajectory have flute. So someday, if we are successful, we can cause the fly to mate with a banana. Then it's really, really successful. Yeah.
So in terms of this coordinate expression of the surface molecule, how the regulation of the gene expression being controlled in the same cell?
Very complex. We dabbled a little bit into it. So it's very combinatorial. Every molecule of cell surface proteins are controlled by multiple transcription factors. And every transcription factor regulates many molecules. So it's both convergent, divergent. So we gave up. After, we have a very good study to show that. And after that, I say, forget about transcription factors. It's not going to simplify our life.
Do you ever see expression of these molecules in glial cells?
At least some of the tellurines, mammalian tellurines, are expressed in glial cells. 10-4, in fact, there is a study saying that it's important for myelination. But in flies, they are mostly enriched in the neurons. You have a question, Imai?
I have a question regarding the Drosophila olfactory system. Yeah. You showed that it's a combinatorial, and you showed that the 3 principles that can rewire the circuit.
Yes.
But that seems to be very different from the situation in mammals, where just a change in receptor is sufficient to create a new projection circuit. So do you think that the 3-step?
So in mammals, as you know, we know a lot, thanks to your work. We know a lot about how ORNs target to glomeruli. Yeah. But we know very little. What are the substrate? Mitral cells, whether they have any, you did some work now recently, but whether they have any, to what extent do mitral cells contribute to this thing, right? So I think, yeah.
Yeah, so my question is, do you think that kind of a three-step mechanisms are also happening during the evolutionary rewiring processes?
Evolutionary rewiring. I think probably they will be play important role in evolutionary rewiring within, insects. But it's we know that mouse olfactory system and a fly olfactory system, even though they look alike, they are not descend from common ancestors, right? They are convergent evolution. So my guess is it's not going to happen between fly and mouse.
Okay. Thank you. Question in the back? I think I will ask maybe the last question.
Yeah.
In the mice, you showed that the tenerin-3 and the
latrophirin 2.
Yeah. Is also expressed in the striatum area.
Yes.
Did you look at the
yes. I mean, that's the part which actually I skipped. Very interesting. They have counter gradient. Once tenerin is dorsomedial, latrophirin 2 is ventrolateral. They're expressing striatum, in pallidum, in cortex. So 10-3 to 10-3, latrophirin 2 is used for cortical, striatal, and striatal pallidal projections. Just short summary.
I see. Maybe it's also used in the basolateral amygdala and central amygdala because
potentially, it's too small for us to look.
Okay, let's thank Liqun again.